Two of my students, and with Mihai Pop, so the two students and Mihai are all in the audience now, so detailed questions are probably going to end up going to them. So Nam and Siavash and Mihai, wherever you are, there you are. Okay. So you've already heard talks about this, but this is basically you're given some short reads in shotgun assembly and shotgun sequencing, and you want to figure out what you're looking at, right? So you could classify at the level of the species if you're lucky, and if you can't classify at that level, then perhaps at the genus or at the family and so forth, going up towards the root of all life. So there's two basic questions, and they're not the same question, although they're related. And one question is, if I just give you a short sequence, what is it? You know, and so you might want to ask what species is it coming from, but you also might want to say what gene is it coming from. And those are two different questions, right? But that's just given a short sequence, what are you looking at? And then the other question is you have a data set, and it has a collection of short sequences, and they come from throughout life, perhaps, and from different genes. And you want to be able to talk about what's in that data set. And of course, then there's the next set of questions which have to do with what are these things doing, but I'm not going to address that kind of question. Right now I'm just going to talk about, given the sequences, what are they? And given a set of sequences, what does the set look like? So what I'm going to describe for you today are two different problems and tools for these two different problems. And the first one is called CEP. And it stands for Sate-enabled phylogenetic placement. And it's a tool that we are using to do taxon identification and phylogenetic profiling. But CEP has its own use, and so I'm going to describe CEP first, and then I'll describe TIP, which uses CEP. So I'm going to begin with multiple sequence alignment. Multiple sequence alignment is not the same thing as what we've just been hearing about from Daniel, which is where you have a sequence and you have a genome and you're mapping that little read to that genome and finding where it is in the genome. It's a different problem where you have a collection of sequences and they all evolved from a common ancestor, and you want to put them into an alignment which reflects that history. So multiple sequence alignment is the tool that we are using to do taxon identification. So it's actually a different algorithmic approach, and I actually don't know how they would compare it to Daniel's approach. It'd be interesting to look at that. So we're going to describe what we're doing for multiple sequence alignment approximately, and then how we're using that inside CEP and then inside TIP. Okay, so multiple sequence alignment, and then how you use that for aligning data sets with fragmentary sequences. So what do we mean by alignment anyway? You have a sequence that evolves into another sequence. So this sequence evolves into this sequence by deleting this substring and changing this T into a C. So it's an evolutionary process, one sequence evolving into another, and you put them into a pairwise alignment to reflect that history. So this is a phylogenetic alignment that reflects the evolutionary history. So true homology, not something you're inferring on the basis of structure or something like that. So for example, because this is deleted, when you align it to the bottom sequence, it's over dashes. There's nothing in the bottom sequence that it actually has a history with, okay? And similarly, this T and this C have a common ancestor, which is the T, right? So they're in the same column. And this T was inserted, so it has nothing up here that it has a common history with, so it's below a dash, okay? So dashes reflect that there's nothing in that sequence that actually has a common history with, and otherwise being in the same position means common history. And so if you have a tree, and you've traced the evolution of all the sequences, and you know the true pairwise alignment on every edge because you know the true history, then transitivity gives you the multiple sequence alignment. So that's the concept of phylogenetic alignment. So if you want to get a tree, the first thing you do is align the sequences, then you take that alignment and you infer the tree, right? That's the standard approach. It's not the only thing you can do, but it's the standard thing to do. So what happens is that it turns out alignment has a big impact on tree estimation. So if you first fix the alignment and then infer the tree, it turns out that your alignment error introduces error into the tree estimation. And you can quantify error by looking at the bipartitions induced by the edges of the true tree and how they correspond to or don't correspond to the edges in the inferred tree. So this is the true tree, this is the estimated tree. The true tree is rooted, the estimated tree is not rooted. You just look at the bipartition, so you don't worry about locations of roots. And the bipartition over here that splits S1 and S2 from the rest exists here, so it's a true positive. And you don't have to mark it, it's not an error. On the other hand, that this bipartition, which splits 4 and 5 from the others, does not exist here. So it's a missing edge or a false negative, and this is a false positive, okay? So that's how you quantify error in trees. And if you do a simulation study, you can look at the estimation error in the alignment, estimation error in the tree, and you can look at different ways to do each step. And for large data sets, we use maximum likelihood because it actually can run on large data sets and can give good results. If you try to use a Bayesian MCMC method, you're pretty much restricted to small data sets. Use something else, you may not get quite the same accuracy. But what about the impact of the alignment estimation? Okay? So we're going to look at simulated data on 1,000 taxon trees with different alignment methods, always followed by maximum likelihood. And this is what we got. This is a study we published in 2009. These are 15 different model conditions. As you go from left to right, they generally get harder. And what's getting harder is you have a higher rate of evolution. Now, it's not just scaling up the rate of evolution, but basically you go from left to right, more evolution. This is maximum likelihood on the true alignment. So in the true alignment, you start off at 5% error, and you end up at 10% error. The rate of evolution goes up, the tree estimation error goes up. But if you have to estimate the alignment, the error goes up much more. So the estimation techniques that we're comparing are Clustall, Mussel, Maft, Prank. Nick Goldman in the room? He already knows these data, so he doesn't care. And then the true alignment. And what you can see is that this method is doing the worst. That's Clustall. Now these are DNA, these are not proteins, okay? The other methods are basically doing about the same, but none of them are as good as the true alignment. Okay, so estimation error in the alignment down here corresponds to estimation error in the tree, and Clustall's not doing very well. That's the basic take-home message, but nothing's doing very well under high rates of evolution. So multiple sequence alignment is basically one of the grand challenges, and it was identified in a National Academies Press thing as one of like the, I don't know, the 6 most important big challenges going on right now. Not just multiple sequence alignment, but also, you know, pairwise alignment, but sequence alignment is really hard. And everything anyone's looked at is NP hard, but that's not the point. The point is that there's poor performance in practice, and rates of evolution and heterogeneity are impacting the error. One of the things that's really interesting for me is data sets like the 1,000 Transcriptome Project, which is a plant transcriptome data set with only 1,000 species, but because of the multi-copy genes, you actually have hundreds of thousands of sequences in individual data sets. So transcriptome data, often very fragmentary. So there are these data sets we're trying to analyze because we're part of this project where there's 100,000 sequences and something like 2/3 of them are short. So it's a mixture of full length and short sequences, and how do you align them? This is the histogram of sequence lengths. So this is crazy, right? This is the P450 cytochrome data set, and this is not sort of typical kind of stuff we're used to dealing with normal data, right? This is really, really, really nasty data. Okay, so how might you try to deal with a data set like that if you're trying to do phylogeny? Well, you might take the full-length sequences, get an alignment in a tree on the full-length sequences, and then take every remaining sequence and insert it into the tree, right? That's called phylogenetic placement. Now, phylogenetic placement was originally introduced as a way of dealing with metagenomic data, but it's not only for metagenomic data. So I'm introducing this problem, phylogenetic placement, via multiple sequence alignment and via how do you deal with fragmentary data. So in this problem, imagine you have all of your sequences are cytochrome, and some of them are short and some of them are long, and you get a tree based upon the full-length sequences, you get an alignment based upon the full-length sequences, and then you take each remaining sequence and you put it into the tree somewhere. Okay? That's the idea. How do you do it? So this is the problem. The input is a tree and an alignment on your full-length sequences, and you also have these short sequences, which you call query sequences, and you want to return the placement of every additional sequence into that tree. First step is you align every query sequence to the backbone alignment, which is the sequence alignment you have on the full-length sequences. The second step is you use that alignment to place the sequences into the tree. So this has been approached in several different papers. In fact, Nick, you have a paper on this. And so the first idea of how do you do that alignment, HMM Align, which is part of the toolkit out of Hammer, will build a hidden Markov model based upon the backbone alignment and then use that hidden Markov model to align every remaining sequence to it. And once you've gotten that alignment, you can then place it into the tree. Then there's something called Papara by Berger and Stamatakis, which is actually using an inference of ancestral sequences in the tree to do the alignment. So anyway, the first step is for each query sequence, you're going to independently align them to the backbone alignment. And then the second step, once you've gotten that alignment, however you got it, you're going to then use maximum likelihood to place it into the tree. The two tools for doing maximum likelihood are pPlacer and EPA. They basically have exactly the same optimization criterion. They have the same accuracy, and the only difference is running time and bells and whistles. So we used pPlacer because it had a better interface for us to work with. And we did a simulation study to evaluate those two techniques, HMM Align or Papara. Those two techniques have actually different algorithmic designs and so potentially different performance. And because we had already seen what happens with rates of evolution impacting alignment accuracy, we wanted to see what happened with these two methods for inferring the alignment of the query sequence to the backbone alignment. So this is the result of the simulation study that we did with 3 different model conditions. Each one has a model tree with a backbone alignment, which has 500 sequences in it, and we're using the backbone alignment to infer the alignment of the query sequences to the backbone alignment using either Hammer or Papara, followed by pPlacer. And the y-axis is the error, and it's the placement error, how far you are from the correct location in the tree. And as you increase the rate of evolution, this is a low rate of evolution, medium, and high, the error rates increase. And what you can see is that under low rates of evolution, both methods are really good. And under high rates of evolution, Papara is worse, right? So Papara is having a higher increase in the rate of error with rate of evolution, which is in some ways not surprising at all. It's trying to infer ancestral sequences in order to place sequences into the tree. The inference of ancestral sequences is hard. The other one is just building a hidden Markov model and using that to align the other sequences. That is also impacted by the rate of evolution, but not as badly. Okay? Now, I don't know why Hammer's ability to align is impacted by the rate of evolution, but it clearly is. Okay. So what is Hammer plus pPlacer doing again? It takes the backbone alignment, it builds a hidden Markov model for the backbone alignment, and it uses that to align the remaining sequences, and each one gets added into the tree. So there is one hidden Markov model for the entire alignment, right? And this works well when you have a low rate of evolution, and then it starts to degrade, okay? Why? I don't know why. But it does suggest that maybe instead of using one hidden Markov model, you should break this into 2 pieces and take the same backbone alignment, but now build a hidden Markov model here and a hidden Markov model here. Now you have 2 hidden Markov models. And now you take each of your query sequences and say, which one should you align it to? Whichever one gives you the better score, that's the one you align it to. Okay? So once you add your query sequence into the backbone alignment based upon this subset, by transitivity, you actually get an alignment of the query sequence to the whole backbone. Okay? So you actually don't have to worry about it. But maybe 2 isn't enough, maybe you should do 4. And maybe 4 is not enough, maybe you should do more, right? So we did an exploration of this, and we published this a few years ago in the Pacific Symposium of Biocomputing. And so here's one of the simulations we did under a low rate of evolution. So just to give you a context, this is running time, and this is error. So what you want to be is down here, right? Now, under a low rate of evolution, hammer followed by p-placer has pretty good error. Papa-Ra followed by p-placer has lower error, but it takes more time. And then this thing, which is one of our techniques, has error that's somewhere in between the two, but takes more time. So these are two parameters you'll see. It says 50/1000, 250/1000, 1000/1000. There's two parameters for this. One is, the first parameter is how many sequences in your alignment subset. So when you're basically breaking the tree into subsets, how small is the smallest subset? The second one is, once you've done the alignment, you're allowing yourself to insert into the tree with a certain subset size. So two different parameters. The main thing is that from this figure you can see that it's really hard to do better than hammer plus p-placer or Papa-Ra plus p-placer in terms of accuracy. This one has an alignment subset size of 10. You're breaking the tree into 50 subsets of size 10 each. Okay? That gives the best accuracy, but then you allow yourself to place into everything. Low rate of evolution, it's hard to beat those other methods, but you can. With a high rate of evolution, it's really easy to beat them. So here's hammer plus p-placer, here's Papa-Ra plus p-placer, and all of these versions improve upon both in terms of error, okay? Maybe not in terms of running time. So both of these algorithmic parameters are impacting accuracy. So we realize that small alignment subsets improve alignment accuracy, but they increase running time. A big subset for where you place it improves accuracy, but increases running time. So we came up with a rule of just do 10%. 10% alignment subset, then just insert there. And this is what we got using that 10% rule in red, okay? Our error also increases with the rate of evolution, but not as much. That's the main thing. Now this is under 3 different rates of evolution, okay? Low rate of evolution, we're in between Papa-Ra and hammer. High rate of evolution, or medium, we have better accuracy. Now on a biological data set with a relatively low rate of evolution, this is the running time, this is memory usage, and this is error. So they're all having low error. We actually have the highest, but these are all very low. This is like half an edge off from where you want to be. But running time, big difference. Memory usage, big difference. This is 75 gigs or so, 65 gigs, and ours is down here. Okay, so this is just phylogenetic placement. Now I'm going to switch into doing taxon identification, okay? How do you do taxon identification? I've shown you how to do phylogenetic placement. I have a tree, I have an alignment, and now I have a short sequence, I can put it into the tree. Can I use this directly to do taxon identification? So if you're going to do that, you have to have the sequences you're dealing with all coming from the same gene. You can't just do it naively. So the two basic questions are, what is this fragment? And what is the set of fragments I'm dealing with, right? So the first question is, what is this fragment? So the idea to use CEP is that you're going to take the fragment you have and you're going to figure out the gene it comes from. And we've already heard that that's not a trivial problem. So you take your fragment and you say, can I predict what gene this is coming from? And if I can, then I'm going to use it. The second thing is, for the whole set, what do I do? So first I'm going to tell you what we do with the fragment, the first problem. So you determine the gene for your fragment. If you recognize what gene it is, you go out and you get full-length sequences for that gene, and now you have a phylogenetic placement problem. Okay? Easy, right? So we did that, and CEP did a really good job of placing things, except that it would almost always characterize every fragment at every taxonomic level, species, genus, etc. And if you don't want to make false predictions, CEP was not very good. CEP was always going to give you a prediction, okay? So what we did is we combined the output of CEP with statistical support. So when you do the alignment, you get alternative alignments from the different HMMs. Each one has some confidence. you also get, for every alignment you get alternative placements in the tree, each with some confidence. So if you take all of that into consideration, you can say, give me all the placements inside the tree that give you enough statistical support. And you can have that as a threshold. And then you move up to the LCA, back to that idea that Daniel brought up, to the most recent common ancestor of all of those placements. So now you've got this parameter that you're able to tune, how much support you require, and that pushes you up away from the leaves, okay? So they can reduce the false positive rate, but you're giving up some of the true positive. So we compared this for taxon identification just of the fragments to methods, some of which can classify everything. So Fimbul can classify everything, NBC can classify everything. Metaphylar is, again, a marker-based gene way of doing this kind of classification. We use the same marker genes that Metaphylar uses. And so we're basically comparing Metaphylar and TIP, which are both marker-based, to Fimbul and NBC, which are not marker-based, okay? And this is what we got in a non-leave-out experiment from data that are generated by the 30 marker genes, and then we introduce error into them, 454 error, so we have indels. And so this, if you just look at this level, this is the default amount of error in the 454, and you're saying if you have species and you've introduced the error, how well are you doing it? Green means you make a correct classification, red means you make an incorrect classification, and everything else is you make no classification. So one thing you can see is Fimbul never makes a classification at the species level, okay? Fimbul will not classify at the species level. You have to move up to genus before you can talk about Fimbul. But TIP is classifying more at every level than the others. And at this level, it's making some false classifications, not too many, and more true classifications than the others. And the same thing is true basically as you move over here. Okay. If you go to very high amounts of 454 error, TIP is actually pretty robust to these indel errors. It's still making classifications, and most of them are correct. Okay? But this is a non-leave-out experiment. Here's another non-leave-out experiment but with Illumina error. And now everything is pretty good at the Illumina error model. Fimbul perhaps not as good as Metaphylar, and NBC not as good as the two marker-based. Okay, so marker-based analyses are doing well for classifying things that come from the marker genes. That's all we're saying here, right? Because it can't classify something if it doesn't come from the marker genes. This does not evaluate how well you can classify things that come from other parts of the genome. Abundance profiling is the other question. Abundance profiling is you have data and you want to know the distribution according to some taxonomic level. For example, species A through E, this would be your distribution. This might be the true distribution. And then you have an estimation of the distribution, and as we saw, you might predict that there's 200 species when in fact there's only 20, right? So it's how accurate is your estimation of the distribution at some taxonomic level. That's the mathematical question here. What I want you to realize here is that you don't have to classify everything to do this well. If you only classify 10% of the sequences, but you do those correctly, you might do this well. You'll have time to ask questions. So we compared Fimbul, NBC, Metaphylar, and Metaflan. Metaflan is another marker-based analysis. And again, we're only using the 30 marker genes that Metaphylar is working with. And we worked with this collection of simulated data. So what these are basically biological data into which you've put in fragments and with errors, and where you know the distribution of the species. Okay? And there are different sequence lengths. Some of them are short, some of them are longer. And we're basically saying short fragment data sets, the average length is at most 100, and the long ones go up to, are the longer ones than that. Okay, this is the species level for, if I remember correctly, these are all the long fragments. And at this, what we're measuring is the error in the distribution, and the root mean squared error in the distribution. And so low is better than high. So closer to the bottom is better. And these methods across these 5 different data sets, blue is always having the lowest error, that's TIP, and we're using BLAST to do gene calling. Okay, and we're only classifying things that map to the 30 marker genes. So this is just at the species level. This is just the long sequences. This is the overall stuff we saw on short versus long for the different data sets at the different levels. And I think one thing that's interesting to see here is that, well, first of all, Fimble doesn't classify at the species level, so you get nothing there. There's different performance based upon whether it's short versus long. For short sequences, the best performing, I guess we're doing the best across these levels, and we're pretty close to the best, Metaflan being the other one at the species level. And Metaflan's good here again. Okay? Long fragments, Fimble turns out to be good at the class level. So there's different methods have different performance at different types of sequence data, short versus long. So one thing that's interesting is that the things that are doing the best here basically are the marker-based analyses. So they're only classifying a subset of the fragments, but they're able to classify those well. So some observations. TIP and Metafiler and also Metaflan are methods that use marker genes, and they're actually only classifying a subset, and they will fail to classify anything that doesn't match those genes. They're both more accurate than Fimble at classifying fragments from the 30 marker genes, and that's not surprising. They're designed for those 30 marker genes. Nevertheless, it's interesting. And most methods are very much affected by sequencing errors. This only has to do with classification of fragments, not profiling. They're really impacted by indels. So to the extent that your sequencing technology is introducing insertions and deletions, methods that are based upon k-mers are more impacted by error. So you have to be able to deal with indel error if you're going to work with sequence data that have indels. In terms of taxonomic profiling, the marker-based profiling actually is very, very good, even though it's only classifying a subset of the data. And of course, the question is, which marker genes should you work with? And we're using the same group of genes that Metafiler is working with, and that's a different set than Metaflan. So why is this working so well? I think it's the multiple sequence alignment that's actually helping. The fact that you're working with a whole set of sequences and getting an alignment on that set is the key to this working. It allows you to deal with indels better than just working with k-mers. So with that, I will take questions, but I just want to, the students who worked on this, they're in the audience, but now you can recognize them. This is Siavash, and this is with short hair, and this is Nam, and Mihai, who's working on our testing of all this. Thank you.
It's kind of the same point I was making the talk before, but so in human genetics we do human ancestry, and there it's very, very hard to, among similar groups, you can't define any bit of the DNA accurately. Okay, but using the whole genome of data, you can say this guy looks like a mix of this and that. Okay, despite the fact that all of the data is noisy.
You're talking about doing a mixture model.
Essentially. So
yeah,
if you do a mixture model which takes into account the uncertainty of the data, you should be able to tell the difference between something with 700 species and something with 10. That should be, that should be possible. And as you said, that's the sort of thing we should be discussing.
Well, so in some sense, we are working with a mixture model in TIP by saying that our, well, it's not really a mixture model, but it's close. We're basically saying we have uncertainty about where this sequence is coming from. Is it coming from this gene or from that gene or from that gene? Is it coming from this species or that species or that species? So in some sense, there's a mixture model there, but not quite. It's more for uncertainty.
But you're not modeling the kind of the total of what you get.
No, but we could. We actually do output the confidence. There is confidence in the estimate that could be returned. There is that. So we could actually give you, with some probability it's this species, with some probability it's that species. We could actually return that as a distribution.
Yeah.
Sorry, I had two questions. The first one, can you comment on, my impression is that Metaflan is good also because it's very, very fast. There's a very small reference
So
data.
this has nothing to do with speed. There's
Yes.
no information here about speed. One thing I should point out here is Metaflan, where it works well, is where it works best, is on the data sets that it was trained on.
Yeah.
So how it works on the data sets it's not trained on, not as well.
Sure. No, I just wondering if you. So I was going to go on saying just my impression with the phylogenetic-based methods is they're rather slow. So could you comment on that?
We have not optimized this for speed at all, but Siavash, you have a comment?
They are slower for the markers they actually classify, but because you have something like 30 markers, it's a tiny fraction of the whole genome. So you wind up working on a tiny fraction of fragments, which means overall they are not much slower than something that looks at the entire thing because you are, reducing your data set.
Yeah, the step one is to say what are you actually going to try to classify, and that's already been a huge data reduction. And then the stuff that you are classifying, that takes time. On the other hand, it's completely parallelized. So where you're spending your time is more time there, but there's a smaller amount of data that you're doing it on.
I mean, my own experience is with one PhyloSift, which is
Which would?
a PhyloSift from Aaron Darling and the ISON lab, and that's a phylogenetic method with a marker gene database. This is Hammer, I think, and P-Placer, so something not.
So this is more like this, that you're talking more like the papers by Stamatakis et al. So the point is here, this comparison is a running time comparison for SEP, which is not TIP, right? But it's close. If you don't break into subsets, running time for Papara is up here, for just a single HMM is here, this is what we are. So breaking into smaller subsets and placing locally reduces the running time. So there's an advantage here that we're getting computationally because of how we do the placement.
Sure.
So it's not exactly, again, the details always matter, and the running time is very much, you can bring the running time down by this technique.
And can I just ask one more question? So relating to the error rate of P-Placer that you showed, which you had for different rates of evolution, I don't understand what M4, M3, and M2 is, but can you put that into context in terms of amino acid divergence or something?
These are nucleotide simulations, not amino acid.
Sorry, nucleotide.
And the M2 is at saturation. So that this is a data set with only 500 sequences in it, but at that level, you will find a pair of sequences that are at or close to 75% p distance from each other, which is what you would get for random data. So where M3 is not at saturation, it's a lower rate of evolution, and M4 is sufficiently below that it's actually easy to align.
Okay.
So these are actually data sets that were published in Science for studying alignment methods.
Yeah, so these are the multiple sequence alignment comparison.
Right.
Okay.
Right. But basically, M2 is at saturation. And some of the marker genes that we're working with are at saturation or past saturation. Okay? So there are lots of marker genes which evolve fast enough that you have to be able to deal with, evolutionary distances where things look random with respect to each other.
How many different species are in this data set?
How many different species are in which data set?
And the ones you use for benchmarking?
For the phylogenetic taxon identification of here, these?
It's
Ma'am?
listed under genome, so it ranges from 19 to
Yeah, okay.
130.
Right there.
These are all from distinct species. These are all distinct species.
Yeah.
Yeah.
Yeah. So these are, with the exception of one, they're all published data sets. For one of them, we took the same data, but just took to get a different sort of rate of evolution.
Oh, these are pretty massive.
You had a question.
Oh yes. Candy, have you tried to consider using assembly contigs, to do it? Do you think the performance will improve?
So that's the difference between short and long. It's basically the difference between short and long. So you can sort of see here the difference between short and long for phylogenetic profiling, right? And so if you look at the long fragments, these long fragments start at 100, more than 100, go up to 900. So this is the error rates. They're not, I don't think, substantially different in terms of the profiling. It's just that which methods are doing well is changing.
Because you got contigs can be longer than 900, and then you will give you more accurate, right?
But that assumes that metagenomic assembly is possible.
Possible, not assembly,
Well, okay, but you, I mean, if you can't trust
Yes.
assembly, then how do you trust contigs? I mean, they're all basically the same challenge, right? So, yeah.
So for did you observe something like huge variability of number of reads which can map to different marker genes? Or do you deal
I don't think we noted that. Did we note that?
didn't note
We didn't notice that. We didn't pay attention to that.
you didn't you look at that, didn't notice, or
Well, that's part of this because the marker genes are single copies. So your prediction of the distribution is inherently that estimate, right?
supposedly, but I remember when I look at the metaflan marker gene, it varies a lot.
But that's metaflan. But metaflan has a different set of marker genes. So they have clade-based marker genes. So for a given clade, you have things that are marker genes within the clade. But they're not going to be marker genes if you take a larger perspective.
Those are the universal 25 marker genes.
The ones that we're working with are the ones that are universal, single copy, and resistant to HGT.
Actually, for the 16 assets, they do show copy number variation. So there's some.
We're not basing this on 16S. So this analysis is based only on the ones that we are using, that we and Metaphyler are using, are single copy, universal, and resistance to HGT. So we're not using 16S. We have tested phylogenetic identification for 16S, and we work well there too, but this is not what we're basing it on. So this phylogenetic profiling is only on these universal ones.
So is that one of the connections that is also used by the Jonathan Eisen's lab?
Yeah.
Yeah.
This is very efficient, only 25 genes.
I think it tells you something about the value of marker-based analysis. Now, TIP doesn't have to depend upon those properties. So we have been able to work with 16S and other things where you get a gene tree, you get a gene sequence alignment, you get a gene tree, and then you can map onto a taxonomy. So all of these predictions are based upon your taxonomy being reasonable. And if your taxonomy is wrong, then these analyses are going to be wrong, right? But you can work with any kind of thing, including multi-copy. You don't have to have single copy. You don't have to have universal to be able to work with it. On the other hand, once you get an alignment and you want to add it to a taxonomy, if your taxonomy is problematic, then you have a problem.
So thank you very much again.
Okay, thank you.